Good evening, everyone. Today I will be presenting my senior research project on the effects of home cleaning products on mobile phone bacterial growth. To begin, I'd like to share a little bit about myself. My name is Villalobos Garcia. I am a senior at George Washington High School, and in the fall I plan to attend James Madison University, where I plan to major in neuroscience. Mobile phones have become part of our everyday life and our daily routines as for its common uses. However, they serve as vehicles for bacteria as they are constantly being carried from place to place, which can play as a health hazard as bacteria can grow and stick to other surfaces. To prevent its spread, my study will compare the effectiveness of home cleaning products and finding alternate safe and environmental friendly paths to minimize and hopefully eliminate bacteria growth found on mobile phone surfaces. Before my experiment was conducted, I reviewed several academic studies pertaining to my topic, the first being contamination. One of the main factors for bacteria contamination is hand hygiene. Multiple studies have concluded that the lack of hand washing can accumulate from bacteria found on any surface to then being transmitted to different surfaces through physical contact. Another contributing factor would be the cleaning frequency, as lack contributes to increase contamination rates, allowing bacteria to grow and stick to phone surfaces. Tying in with that, studies have shown that the back surface of phones are less frequently cleaned due to not being considered as high-touch areas as the touch screen is. Storage and temperature also play a part in bacteria contamination, as warm locations like handbags and jean pockets provide high bacteria contamination rates. Another area of focus were the types of bacteria that were commonly found on mobile devices. Staphylococcus aureus is a type of bacteria and is found in the upper region of the human body and face, and is spread to others by contaminated hands as well as facial contact. Major routes of the skin bacteria transfer onto mobile phones from fingers, facial, and palm contact. Staphylococci can survive for months, building in on itself and being the cause for illnesses such as the common cold. This skin bacteria contributes to the identification of bacteria, but also addressing Pseudomonas, which serves as the leading bacteria after Staphylococcus aureus. The last focus of my research were common household cleaning products. Studies have shown that 70% isopropyl alcohol statistically minimizes germs by slowing down its growth on different surfaces with references to antimicrobial resistance patterns, which is the germ's ability to fight back on disinfectant products designed to kill them. Unfortunately, along with many other cleaning products, it carries potential unseen health hazards, such as serving as a lung irritant depending on the amount used. Bleach and white distilled vinegar are both common household disinfectant products, as its effectiveness in bacterial elimination reduce seasonal sickness peaks, and its familiarity with the ingredients creates a sense of comfort and safety with its users. Tying in with alcohol, products containing vinegar and bleach can cause lung irritation, with the downside of not entirely eliminating skin bacteria on all surfaces. The research question I gathered from reviewing my literature was, how do at-home cleaning products affect the bacterial growth on mobile phones? My research hypothesis states, if DIY solutions are used to disinfect mobile phones, then there will be a greater bacteria reduction than the other products. Whereas my null hypothesis states no significant difference in bacteria reduction will result from the use of DIY solutions. There were two variables in my experiment, which were the main ones. Then my independent variable in the experiment was the home cleaning products I used, with my dependent variable being bacterial colony growth. Tap water was used as my control variable within my experiment, with my constants being the petri dishes used, as well as my participant population all being from the George Washington High School student body. Before beginning my experiment, I obtained consent forms from my participants and parental guardians, in which I resulted with 30 petri dishes as my sample size. For my materials, I gathered phones from my participants, petri dishes, my disinfectant products, which were 91 isopropyl alcohol, 2 do-it-yourself, or also known as DIY, solutions, 1 containing white distilled vinegar, tea tree oil, and the Dr. Pepper soft drink, and the other containing water and baking soda. 7th generation disinfectant spray, and all in all, disinfectant spray were also used with paper towels, sandwich bags, spray bottles, and sterile swabs also being in my materials. In preparation for my experiment, I randomly assigned participants to their groups through the use of a random name generator. The first 6 names would be placed into a group until all 5 groups were filled. In addition, I labeled sandwich bags according to their group number, person order, and whether if it was before or after the disinfectant application. For each group, I collected each participant phones, swabbed each phone with a clean swab before the disinfectant was applied, swabbing the entirety of the posterior surface of the phone for 10 seconds, excluding the camera. The swabs were then tightly sealed into their designated before bags. I then began to prepare for my disinfectant application portion by taking a clean paper towel, spraying the disinfectant onto the towel, then applying it to the same surface. After applying the product to all of the phones, I swabbed each phone with different sterile swabs and tightly sealed them into their designated after bags. The following day, the bags were taken to the institute, into the lab where the swabs were then applied to petri dishes and were incubated upside down for 48 hours at 37 degrees Celsius. After the 48 hours have passed, the petri dishes were then taken out to be counted. At the end of my experiment, I compiled my raw data into an Excel spreadsheet and conducted my statistical analysis. For my statistical analysis, I ran a one-way ANOVA test to compare the different means and bacterial colonies among each different disinfectant group. For my test, I was able to project my null hypothesis at the 0.05 significance level with a p-value of 0.021. Overall, all disinfectant products displayed reduction in bacterial colonies. All in all, cleaner resulted in the greatest disinfection rate of 36% with the lowest rate of disinfection being the 91% isopropyl alcohol, going against what previous literature studies have stated about alcohol products reducing the most bacteria. At the 0.05 level of significance, I was able to conclude that home cleaning products effectively reduce bacterial contamination through sufficient evidence found within my experiment. Therefore, they can be used as alternatives rather than commercial products. The results from my experiment indicate that home cleaning products effectively reduce bacterial growth on mobile phone devices. My study coincides with what is already primarily found, which is that home cleaning products do reduce bacterial growth, but my experiment found large reduction rates on different high-touch area surfaces with its natural and familiar ingredients. The use of home cleaning products can be beneficial as they are commonly found throughout households as well as already being a familiar household name, providing easy access to products as well as being a cheaper option due to most ingredients already being found in homes. Limitations in my study primarily came from my participant selection. A larger sample size would accurately better represent my population with both participant size and the types of disinfectants used. In addition to my participants, including people outside of the George Washington High School student body would also better represent my population by including a more diverse sample. Using participants in various age groups can help benefit future studies by examining different levels of bacterial growth and determining whether or not the same disinfectants used still produce a high disinfection rate. Testing different device surfaces to which is used more often can as well test different levels of bacterial growth with the same products. Lastly, few studies concluded the posterior surface of a cell phone is the least frequently cleaned surface on a device. Testing on the touchscreen surface of the device can help examine a similar yet different surface area as well as different bacterial growth rather than just limiting it to the back surface. Future research should look further into using a larger population size with diversity in age and location demographics for better accuracy and better representation. Furthermore, more research is needed on the use of entirely natural ingredient-based disinfectant products to ensure environmental friendly alternatives. Different surface areas, whether that be testing the touchscreen area of a mobile phone or a different high-touch area surface. While I still have not mastered public speaking, the presentation portion of my senior research project has allowed me to fully experience public speaking with a big audience while still expanding my knowledge in the science field. My SRP has also taught me the importance of managing my time. Research takes plenty of time and patience, as well as the importance of not leaving things to do last minute. I would first like to thank Dr. Bocock for approving my project, Ms. Long for guiding me throughout the entire process of researching as well as performing my experiment. I would also like to thank Mrs. Dillon for helping me with my materials and bacteria application portion of my experiment. Next, I would like to thank Ms. Agner for also approving my project. And lastly, I would like to thank my participants for allowing me to collect bacteria off of their phones. I will now open the floor to any questions.
You mentioned where someone stored their phone may make a difference in bacterial growth. Did you take that into account as the students handed over their phones when you did that initial swab?
Dr. Mark Jones is asking if I took into account if whether my participants were handing me their phones from their jean pockets or their handbags. No, I did not take into account whether they were coming from their jean pockets. My participants just handed me their cell phones from their hand.
I think from your research, you probably would have.
Yes, I should have elaborated more and searched more into whether.
But you probably would have thought that if I stored it in my pocket, more than likely it's going to have more.
Yes, more bacteria would be found.
Did you swab all phones in the same place?
Ms. Bullard is asking if I swabbed all of the phones from the same place. Yes, I swabbed all of the phones on the back surface of the phone, so their case, and I swabbed the entirety of it, excluding the cameras.
You mentioned that in your research, it showed that isopropyl alcohol would probably have the best results, but in your studies, it did not. Why do you think that was? Why do you think your study contradicted your research?
Mr. Eid is asking why my study differed from the literature I found on alcohol reducing more bacteria. In my literature review, 70% isopropyl alcohol, statistically, that type of alcohol did minimize the most. Other studies have also shown that not all alcohol products, I know there's like 51% or 50% alcohol, and in my study used 71%. I was genuinely just, I guess for including alcohol products into my experiment, I just used 71 isopropyl alcohol to examine how that would also play into the disinfection rate.
Do you think if you used a different percentage, you would have had different results?
Yes, I do think that if I used a different percentage rate, that it would conclude in a different disinfection rate, yes.
I'm curious as to why you swabbed the back of the phone rather than the screen. Could you elaborate on that?
Ms. Ken is asking why I swabbed the posterior surface of the phone instead of the touch screen. In my literature review studies, they have concluded that considering the touch screen is more frequently used, people are more prone to clean that area surface instead of excluding the posterior surface, which can accumulate more considering you're cleaning the other surface rather than the other surface.
In your methodology part where you wiped the surface, did you have a length of time that you let the disinfect maybe do its job before you swabbed it? Or did you find anything in your research about that?
Mrs. Collins is asking if, can you repeat your question? Sorry.
That's fine. Yeah, it was two parts in there. So when you wiped down your surface, did you leave it on there for a length of time before you swabbed it? Or did you find anything in your studies about that?
Mrs. Collins is asking if I left a certain amount of time before from when I applied the disinfectant to when I swabbed it with the sterile swab. I applied the disinfectant, but I did not let it dry into its entirety because you have to have like the wet, damp surface in order for the bacteria to pick up on the sterile swabs. And can you repeat your second part of the question?
Did you find anything in your studies that stated like how long it would take something? Like did it say like a recommended like 5 minutes to let it do its job before, you know, it would kill the bacteria?
No, in my literature review studies, they did not specifically say how long to, or like if it would, I guess, not if I were to apply and leave it for a certain amount of time. No specific time was stated in